And it's my pleasure and honor to introduce our keynote, Larry Smarr, who doesn't need actually introduction. Larry, please.
Thanks very much, Gene. It's very odd that I'm here and you've been looking at my data. But none of this was ever meant to happen exactly. I've sort of stumbled into this, even though I've been gathering this data in an accelerating way for the last 10 years. But nothing makes me happier than to see people making use of it, because that's really, in the end, mainly why I'm doing it. Now, I'm only at 5 slides, so I just want to zero in on the main story here, which is that we're beginning to be able to read out for the first time what's actually going on in our bodies. And I'm going to focus on just one dynamic coupled system, which is DNA, the genes it has, and the human immune system. As you know, I self-diagnosed myself by just looking at biomarkers that I had a form of inflammatory bowel disease. That's an autoimmune disease, chronic incurable disease. And in particular, Crohn's. I won't have time to take you through how I did that, but it was involved looking at biomarkers as well as imagery like MRIs and colonoscopies and so forth. When you look into the literature, this is what the state of understanding of why we have autoimmune diseases. Namely, we don't know. But we know there's 3 component parts. Something is amiss in your host DNA, your human DNA. In other words, you have some variants from quote unquote normal. Then there's a dysbiosis in your microbiome in your gut, which has 100 times as many genes as your human DNA does. And between those two, one more or less stable, one changing every time you eat something, is the human immune system. It trains from birth on your gut microbiome as it develops, so you have a joint rapid development of both the gut microbiome and its diversity and the immune system's sophistication. So I realized that as part of my quantification that I should set out to, if I want to understand why I've got this disease, I should set out to quantify all 3. So the first easiest was to do 23andMe. That's now under $100 and just takes a little spit. I'm working with them now on creating an IBD advisory board. There are 200,000 people like me. How many of you have done 23andMe, for instance, right? Okay, well, there's 200,000 of you, and it'll be 1 million within a year or so. And so within this vast database, each of the people that do it get 1 million SNPs. And that's where 90% of human variation occurs. When you put in Crohn's into 23andMe, this is the chart you get. So each of those is a SNP, which is associated with a malfunction of some gene involved with the immune system. And the normal ones, where I'm green, meaning I'm less likely to have it, is the typical Crohn's, which is ileal Crohn's, the end of the small intestine, which is really very disabling, and I'm sure many of you know people that have that. Whereas what I found out is I'm actually on the interleukin-23 receptor gene, which is on the first chromosome, and that is at the heart of the cytokine network that's involved with the T cells that is really at the front end of your immune system. So what it says is that I tend to be more pro-inflammatory in that particular thing, and there's a whole cascade that comes from that. And the thing that I found very effective is I didn't know what interleukin was. And so I set out to educate myself, and the way you do it is through PubMed, and you read hundreds of medical research papers and figure it all out, and then there's one. And so it's actually in the heart of regulating Crohn's disease. Well, I've got my full human genome sequenced now. I'm on the advisory board of Complete Genomics, as well as and George Church is there with me, and then I'm through that working on the Personal Genome Project at Harvard Medical School. So that means that we can look at rare variants, we can look at things that are much, because the SNPs are 1/10 of 1% of the human DNA. But the point is that already is something as simple as putting a little spit in a tube and putting Crohn's in a search bar and then doing a little Google, you're already at this level of depth of understanding why you might have this disease. Remember, I didn't know this until I was 60. There are only 5% of people that have Crohn's that are diagnosed after 60. So that was pretty easy. So the next one was harder. That involves taking multiple blood tests. And when because I'm a scientist, what I set out to do originally was to try to understand a particular ratio, namely the omega-6 to omega-3 ratio of essential fatty acids in my blood, to figure out how I had de-inflamed my body by getting rid of most of the crap that you eat in our country, and instead tuning it back to what our hunter-gatherer ancestors that developed our genes had, which was more like a 2-to-1 ratio rather than a 21, which the average American has. That's why they're all full of chronic disease. But in doing that, I began to try to dig into subsystems in the body. So I started with things like the glucose insulin system, and then, you know, your liver, your kidney, all these things. But in particular, by taking stool samples, which I've never had a doctor until very recently ask for, and even then, I get one variable that's any use out of what my doctor's asking for. And by the way, I have the best, probably, clinical researcher in IBD in the country as my researcher, so it goes downhill from there. But if you just go to the web, Your Future Health, get a little stool kit. I've done 40 of them now, and pay for it yourself. Then you find out that because your colon is your largest immune organ, you can get lysozyme, which is a direct readout of your innate immune system. It's point defense in the colon wall. And then secretory IgA, which is your most common antibody. And you can see, it's very interesting, the green is the healthy. These are not the same graph. And that's because your immune system has all these subcomponents, because there are all these different threats you face that the body has evolved to customize. So you and sea urchins or something share lysozyme. That was invented a long time ago, evolutionarily. Whereas the adaptive part with antibodies and everything it's much more recent in evolutionary time. Well, this basically bores holes in the side of gram-positive bacteria, but it doesn't do much to gram-negative bacteria. So the thing that you'd want to say is, well, why is that oscillating up and down with a scale that's about a period of say, 3 months? Well, it must be some gram-positive bacteria that the body doesn't like, and so it's activating lysozyme. Well, what would those be? So you're led logically, if you think scientifically, into why you want to test something else. And so that's why I got into, as Jerry mentioned this morning, working with Craig Venner's institute to actually sequence my gut microbiome. Now that's, unlike the human one, which somebody else did that out of, presumably it was CGI, but I don't know. This, actually, we did hands-on. So the Venner Institute did, you basically, you send on dry ice a stool sample, which I just recently did, some more samples, and a couple months later you get a FedEx back with a 2 terabyte disk drive and a 35 gigabyte file. Now at that point you have to have a software guru who has developed a pipeline that people like Ilke help create the software for, of the pipeline that actually does all of the bioinformatics that takes 230 million short Illumina reads, each 100 bases long, and then translates that through, in our case, a couple 100,000 CPU hours of supercomputer time into the relative abundance of, say, 2,500 different species of microbes across 7 phyla or so. And when you compare it, we then downloaded from the Human Microbiome Program the raw reads from, well, 100, but we selected 35 of those healthy people to go across this curve you have of the percentage compared to 100% of the Bacteroides and the Firmicutes. So we have that as a healthy, and that's the average up there. And you can see that the Bacteroides are the dominant one with the Firmicutes. So that's mostly what people think about with little trace elements. But when you look at the Crohn's, and this is just a few Crohn's patients, and same with ulcerative colitis, because that's all that has been done at this level of depth of sequencing in the country that's available online through the Human Microbiome Program. You can see that you have a complete collapse of the blue sector. Well, if your good microbes are all gone, then one of the jobs they have is keeping down all the bad guys. Now, if you look on a log chart of all this, you can actually look at all these traces out to hundreds of, the most abundant microbes, and you find out that the ones that crop up in me are all rare microbes that are there all the time. But ecologically, they're not kept down anymore, and so there's a bloom of these, some of which are pathogenic. And you can see that here, the proteobacteria, E. coli being the most famous, is the big bloom that happens there, whereas the balance between Firmicutes and Bacteroides is still sort of composed. When you look at me, and this is about a year ago, you can see that I would say probably I've got Crohn's, but it also shares the properties of ulcerative colitis and having that. It turns out when I went to Claire Fraser, who actually did the work on the Crohn's, these are all folks with ileal Crohn's. Okay? Whereas what I have is left-sided sigmoid, about 6 inches of my sigmoid. Those are very different diseases. There's there's at least 2 orders of magnitude in density of number of bacteria per cubic centimeter in the end of your small intestine compared to your large intestine. Well, what happens when you look at a time series, so here it is, the one you saw is in the middle. This is where my, say my CRP was 30 times normal in my blood, and my lactoferrin at one point was 125 times the upper limit for healthy, which is one of these measures. What you can see is that there's a 90-fold reduction in fusobacteria. There's a 45-fold reduction in the methanogens. These are archaea, actually third kingdom of life, which people don't even normally think about. I was 20%. And, but what you end up with is the green, E. coli. I'm 10% E. coli. Now that's 150 times what a healthy person would have. And so I'm trapped in an equilibrium far from healthy. And so then, and that's simply because if you had any ecological dynamic model, you'd know that you end up with a multiple equilibrium solution space. And so I'm over here in some other solution space. And I think this is one of the people like David Relman, who's the top microbiologist thinker at Stanford, when his most recent papers are all the tools we're going to be able to borrow from ecological theory and ecological dynamics into medicine to really understand this, because it is an ecological system. So that's just a very quick overview. I mean, there's zillions of other things I've done as well, but just I think that's in the end probably the most central piece. My fondest hope is that I can transition from being a gentleman scientist, doing this stuff on my body. Is a long tradition of that. But it'd be really good if we had people that actually knew what they were doing, that were engaged with this sort of work. And so that's why I'm working with people like John Madison at Kaiser Permanente and others, Mayo Clinic, Mount Sinai, and a number of others. Because the idea is to get this quickly into clinical trials where we can try out this reading out of people in a double-blind clinical trial and really get some science out of it. So that's what you'll see happening, I think, over the next year. Let's do some questions.
Okay, so let's have a question. We have half an hour. Please go ahead. Okay. So then.
Hey, Larry, I'm curious, what do you see maybe 10 years out as your vision of how this might be operationalized for the community at large? And that's aside from cost factors or cost reductions driven by mass consumerization, et cetera.
Well, first of all, there's going to be massive orders of magnitude reduction in cost of getting the data. Already, if I've gone up to Mike Snyder, and you know, it really bummed me out when I found out there was somebody that had 100 times as much data as I did. And so I said the natural thing to do, well, I'd join him. So I went up and gave him 12 vials of blood and a few vials of urine and stool just for showing I was a real blood brother. But the point is, what that's going to do is to go to the full transcriptomics, proteomics, metabolomics, autoimmune arrays, and now what you're doing from a single drop of blood, you're getting 5,000 proteins. And so I'm meeting with Danny Hillis tomorrow, for instance, at Applied Proteomics as one of the companies doing this. There's lots of others. So first of all, you're going to be able to get vastly more markers for vastly less cost and precious bodily fluids that have to be extracted from you. And then things like uBiome is taking off, at least as a startup, where instead of doing all this dry ice and everything else, you just put a Q-tip across your toilet paper after you used it and put it in U.S. mail. And now that's only 16S, but still that will give a pretty good breakdown of things. So I think that's the first step. Then the real question, because this is a completely disruptive moment, as I've been through many before in other industries and other areas, you can't predict through it any more than you can predict through a turbulent, if you're going down the Grand Canyon. I spent 2 weeks going through 200 rapids, and you can't predict where you're going to come out. And so I think whether the consumers, through smartphone apps and things like WellnessFX for blood test, or Inside Tracker, or a number of which are already out and going that will do this blood test, track it, graph it at startup prices. Whether that overwhelms the medical industry, or whether the medical industry from within can reform itself fast enough to get on board and provide some leadership. We'll just have to watch the movie. But that we end up with a population-wide set of data, genetic, biomarkers, microbiome, I'm convinced that will definitely be here in less than 10 years. Yeah.
What about privacy issues with regard to things like 23andMe? Could that information potentially compromise somebody with regard to job security or insurance coverage?
Sure.
No safeguards?
We live in a lawless land, full of lawyers. What could be better? No, I think I think until there's fundamental legal reform, and I think this will drive it, people being infuriated with this. As I said earlier when I was talking to somebody, that I think the fundamental legal reform has to be that any data that your body generates, you own. So in other words, property rights for the data. And then you can lease it to your doctor, you can lease it to insurance companies, whatever. You can set up arrangements. But until that's put in place, it's the Wild West. And people are at risk who what I'm doing is what they call high-risk behavior.
So a popular area for looking at analysis of, say, fecal proteomics is in terms of colon cancer. Would be interesting to know what is the overlap between your data sets and data sets which have been determined for these patients.
Okay, well, I'm giving a talk on that tomorrow at USC in the Oncology and Physical Sciences seminar series that the National Cancer Institute hosts. This blue thing that says 90X is fusobacteria. And as you know, last year there were 2 genome research articles that came out that indicated that there were members of that phylum that are found in much greater abundance in colon cancer samples that are taken out of people. And so then the question becomes, what is the relationship between that particular bacteria, which is generally found in your mouth, in when you get infections down under your gum line and so forth. Now, there's a new theory that was introduced, I guess last year, in Nature, I think it was Nature Reviews in Microbiology, which is the so-called driver-passenger model of microbes in the formation of cancer. So inflammation drives cancer, at least particularly colon cancer. Now, good. What's in between those two words? And so what happens is when you get down to, you can't really, I think, understand this by just getting down to the species level. You have to get down to the strain level. So right now we're evaluating 300 different strains whose genomes are fully known for E. coli. Some of them are if you look at the phylogenetic tree, some of E. coli, some of them are pathogenic and have, through horizontal gene transfer and otherwise, put in virulent genes. And what those do is they're attracted actually to inflammation. So your body's immune system is creating inflammation. That's where the inflammation is coming from. However, it turns out, thanks to Mr. Darwin, that the microbes can adapt with that as a selection pressure to insert genes that allow them to flourish in the presence of, say, antibacterial agents and so forth. But when they do that, they can also get genes which further damage the tissue. And this creates a sort of downward spiral cascade whose end result is colon cancer.
Yeah, I mean, it sounds like a very unequal battle, but then the human species has survived, so I guess we do okay.
Well, some of us, actually none of us survive. You know?
Depends on your time frame. I'm curious, have your reasons for doing this tracking changed over time? What were they when they started and what are they now? And I'm Peter Karp from SRI.
If anybody had told me I was going to be up here 3 years ago, I would have gone somewhere else. No, where I started was back in 2000 when I first left the center of the obesity epidemic in the Midwest and moved to La Jolla and looked around and realized that if I didn't clean up my act, they were going to ship me back. And so I literally started figuring out, well, how could they be so thin and healthy and I could be so fat and slobby? I was like 30 pounds more at least. And I hadn't thought about it very much, so I started now as a scientist, I've read 30 or 40 books on this subject and found out that the body actually has biochemical subsystems in it. And it has biochemical subsystems in it, like the glucose insulin system is perhaps best known and its relationship to storing energy in fat cells. But the eicosanoid system is directly driven for pro-inflammation, anti-inflammation by particularly omega-6 versus omega-3 essential fatty acids. And therefore, you can invert from the actual structure of your body what you ought to be putting in your mouth. And it turned out that that was like a few percent of what Americans are presented with as food and drink. And so I gradually started tracking more and more and getting healthier and healthier, doubling and tripling my strength and losing lots of weight, and never expected that rise. So going from, say, standing on the scales with, that's one number that quantified me in 2000, to say by 2005 doing 150 variables in my blood and stool, to 2008 when I got up to 1 million, and now I'm at 25 billion bases, say. But I never expected to find out that I had a chronic incurable disease. I mean, you could have never you never told me this was going to be the outcome. And it took me 5 years of detective work to go from seeing these odd numbers in which, if I look on my wall at all 150 as time graphs, and you'll just pick out right away the ones that are crazy. And, and that gradually came down to the immune system variables. And I said, what the hell is that about? And so it was really then digging into this scientific detective story and then self-educating myself by reading zillions of things and then hanging out with people like, Lee Hood, who I've worked with for 25 years, and George Church and Mike Snyder and so forth, and having them educate me as well. And then it became a really cool science problem. So that's sort of where I am. It's a really cool science problem. Because there's not much I'm going to do about myself unless I can't change my DNA. I mean, I'm genetically predisposed for the rest of my life unless we do gene therapy. But it is a wonderful, I haven't done this much hands-on science since probably I was a graduate student. So it's very rewarding intellectually. And then giving these talks, I've never had the reaction from audiences when I talked about optical networks and supercomputing and black hole collisions and relativistic astrophysics that I get from any one of these talks. These people come up to me and say, "Well, my daughter's got Crohn's. I've got this, got this." I mean, it's an extraordinary deep human engagement, and for me at 65, that's novel. I was a geek all my life, so.
For me, let me make a comment. For me, one of the neat things about this work, and it comes out in both your paper and in the Mike Snyder paper,
Yeah.
is watching disease evolve in unexpected ways. So imagine that we had to debug computer programs by adding one print statement per month
Right.
to the computer program, right? Yeah. That's not quite how we debug. We put lots of debugging statements in and instrument the programs highly to try to figure out what's happening to this complex beast.
When I started with paper tape,
yeah, right. Me too, actually.
it was sort of like today in medicine.
Right. So the idea that we can so one question is like, what's the right time granularity to be doing this
Well,
instrumentation?
absolutely. And so the whole point to these kind of graphs, which are, by the way, unless somebody can inform me differently, better than anything in the medical literature, which is absurd.
You mean more granular?
They're finer in time. And of course, these are only 2 of 150. And, that's nuts. So I don't know what people have been doing with their time, but they could have been doing this. And when you do this, you see, like any coupled nonlinear system, it sets its own time scales on different variables. So I used to do general relativistic magnetohydrodynamic accretion onto black hole simulations, right? And there would be these natural time scales that came out from the magnetosphere and all this stuff, right? And you can see there's almost, this most recent point over here is May 1st. So I'm clearly going back up into a very high state of inflammation. And so as a result, I'm going to more finely in time take samples. So in early June, I'll take another sample just so I don't miss this. So you see, once you begin to do this, it feeds back on the experimental design. Right? And I'm also just finished talking with NYU, professor there in immunology, and I'm going to send him some blood so he can look down at the T cells, T regs, T helpers, all this stuff, and begin to get to that level of down at the very fine, the root hair level of the immune system with this phenotypical profile as a backdrop. And so I think that's where we're going with this. And furthermore, while it's fun to watch yourself in disease, it would be much better to watch yourself while you're healthy and avoid becoming diseased. And that's where we're going with predictive medicine.
Hi, I'm Jan Arts from University of Leuven in Belgium. And actually I had 2 questions. The first one is the first one that was already asked, which is good. The second one is we're moving towards, technologically to more and more using omics-wide
Yes.
Technologies. And I wonder if you have discussed with people the issue of incidental findings. So suppose we sequence someone just to check one particular, to be able to genotype some SNPs or in some disease, and we find that they have a mutation in the Huntington gene, which basically tells you 100% they'll get the disease, period. Have you had discussions with people about the right to know and the right not to know
Right.
of that type of data? Because if we move into this direction, that will happen constantly.
Well, people can have whatever attitude they want. Maybe just because I'm a scientist, the idea of willingly living in ignorance when you could, in fact, have knowledge is an odd way to live. So I would always want to know. There are people who
Yes.
argue passionately on the other side of it, and I'll be happy to let them do that.
Because it also tells you, well, especially if you look at genomics, it doesn't tell you just about yourself, but also about
Well,
your
what
family.
the hell
So that's
is,
the
what the hell is sure in anything in life? Of course, it's a probabilistic statement that you're going to get out of it. What isn't? I mean, if you know exactly where the stock market's going,
Yeah.
you should be doing that. But we don't. If you know what the weather's going to be 2 weeks from now, you can make a fortune for yourself, but you can't. So everything in our lives actually turns out to be probabilistic, and all you're getting is some more data from which to refine your view of life and what you should be doing.
Okay.
So following up on my operationalizing question, I imagine you have contemplated whether we are on track computationally to be able to do this en masse.
No.
We're not. So where do we need to go?
I can't get my genome done now because SDSC, which has the only large memory supercomputer, and I need like hundreds of gigabytes of RAM, is jammed up because there's so little. The great United States of America has one damn machine sitting at San Diego Supercomputer Center. I got to get in a queue and wait for a few months to make a run. Why? Because in the last 9 months, the number of individuals around the world that have been sequencing microbes has added another 1,000 viruses and another another at least 100 E. coli's. And when I put them all in the reference database, it's now too big to fit in the physical RAM, okay, of Gordon. So I got to go to the virtual one, but the virtual one is so oversubscribed that one person can't take the data that's coming from the genome community on the microbe genomes and do their own microbiome for one instant in time.
So can I follow up with the question? So
Yeah.
People know you have level of acceptance on different hierarchical levels of our academic organizations at NSF, NIH, and at all. I'm sure you raised those questions to people in power. How do you see a way out of it? Because it's only one individual we're talking about, and if we take, another person we talked today about, Mike Snyder, and his program, it's going to be dozens.
He's going to really break it.
Yes, within a few months. So
Yeah.
how do you see our way out of it?
Well, actually, and let me just make a general comment about this group here in DELSA. My great hope lies with you. I don't mind being the point of the spear along with Mike. That's sort of what I've done most of my life. But if we don't build a community that is working on all the various developing software tools, sharing those tools, sharing more data, building up a set of results that become so clear to people that even places like the NIH would invest in computing, which would be quite a revolution. This is, to me, the proximate group. And the fact that you have spent today and tomorrow on these emerging issues, I can't do that. I don't have the credibility that you do. I have different credibility, but I got more data than you do. But seriously, and many of you come from different sectors, different countries. And there's going to be a growing movement. So let me just tell you, the Quantified Self organization started in Kevin Kelly's living room with Gary Wolf and maybe one other person 5 years ago. 5 years ago last September. I gave the keynote to the Quantified Self annual meeting at Stanford last October. There were 500 people in the audience, and there were, how many cities? 50, 60, I don't know, cities that now have Quantified Self meetups in 5 years. And what you're doing has that potential. So all I'd say, because I've got to sadly, before long, run to the airport to get to USC so I can give this cancer talk tomorrow. As you continue to talk about things, don't just look at the technical issues. Look at the social organization. We live in a world of social networks. We can amplify much faster. The reason I tweet and Google Plus and Facebook and all this stuff is to try to amplify, is try to accelerate the rate of progress. And all of you have a very important role to play in that.
Hi, this may sound like an IBD anonymous meeting, but I actually have IBD, and I've known this for more than 20 years. Actually, my father's side of the ancestry has phenotypes for 10 generations. My mother's side about 4. So I come from this a very different way. By the way, I had a grant on omega-3s and 6s as well, fatty acid pathways. What was interesting was I lived in Seattle many years, and it was vitamin D pathways. Vitamin D levels plummeting, and also some incidental vitamin A pathways. So I've come across this, and I don't know if IgA is a good indicator, and from the literature at least, it's a very confusing indicator of what goes on. But
well, the vitamin D level is very important, and I, of course, track that. What's your vitamin D level?
it was in the 20s. When I measured it a couple of years ago, I was shocked. I'm a brown-skinned guy, so it's fascinating how many diseases it affects now. That may be a simple answer to some of these things.
Absolutely. No, it's very dynamically coupled into all of this, and people with IBD often have problems with vitamin D deficiency. That is one reason they end up with osteoporosis.
So I've actually developed this cocktail over many, many years. One of them is a fascinating fungus, Saccharomyces boulardii, which has. Oh, so do I. I just want to say, it's there, but I have the fibrillar mutation, soft tissue, and when I did flow for leukemia for a little while, there's actually cluster autoimmune diseases that, of course, fibrillin is chromosome 1, 3, 5, and 15, but. So there may be other things that are involved that are the envirome, if you will.
So I have a question. Do you take vitamin D3?
Yes, I do.
How many IUs?
Well, I've been experimenting. I've been experimenting with multiple things over the last 7 years, but there's a very fine line about the amount of vitamin D and A. I take vitamin A also. So,
but I mean, how many? 10,000, 50,000 a day?
What D?
IUs?
4,000.
4,000. See,
I've actually tuned it. I've experimented on myself as well for many years now.
Yeah.
So.
Well, I take 10,000 a day, and my vitamin D level has fallen from a high of 55 down into 40 while taking that. So, one of the problems is that the microbes, which are critical in vitamin, the whole synthesis and absorption aspect, I believe we will find direct correlation between the bugs that aren't there and this vitamin D deficiency that occurs, because the IBD means largely that you've got a real dysbiosis in the microbes, and those microbes that are missing, the fact they have 100 times as many genes mean they carry out 100 times as many services for your body. And if they're 90% reduced as the bacteroides are, then where are all those services? So it's amazing that we're still walking around.
Yeah, what I'm finding is also the ratio in A and D is important. So anyways, I'll leave it.
Yeah,
Thank
that's
you.
a whole other thing of supplements.
Great talk, by the way.
We could talk about it. Sure.
Short, very short.
Yeah.
Very short.
So are there any data parallel algorithms on the horizon that wouldn't require all that memory in one place, but it could be distributed?
Right. So this is what happens typically, with high-performance computing issues, is that as you come to a new architecture of supercomputer, you rethink the algorithms and you rethink the actual techniques you're doing to get to the answer. So it could be that there's a massive parallel version of this, and that's one of the things that I'm trying to get. I just came from chairing the Advisory Committee on Cyber Infrastructure at NSF for the last day and a half, trying to get them to invest more in algorithm development because I used to have the largest shared memory array and supercomputers in the country, and that would have been great to have for this, but back then a lot of memory was a gigabyte. That was millions of dollars, right? And now it's throwaway. So algorithms are going to be critical. And what do you really care? Do you really care who these guys are? Or do you care about the gene ensemble that they represent? Or do you care about the transcriptome? Which of the genes are actually being turned on? Those are all different measurements. And it might well be that looking at, say, the transcriptome, or even better, the metatranscriptome of both the co-creation of RNA by the human and the microbes, DNA, could be actually telling you more about what's really going on than this taxonomy approach, right? And we have done the full COG and KEGS for those of you who care of this. That's why it took 200,000 or 300,000 CPU hours. This part doesn't take that much. And so that's the next level of analysis. But the other thing is, of course, when you do these reference genomes, there's still a third of the short reads that there's no match to any known microbe. And of course, that's because 85% of the, at least, of the colon microbes are anaerobic and are not culturable by traditional, put them in a petri dish methods. So there's a whole range of new technologies coming from MIMS and nano that allow you in anaerobic chambers to actually get these things up and then use single cell sequencing to get the remainder. But we have a vast unknown set of microbes and their function still ahead of us. Even though they're small in the tail, they can have, as you can see, under the right conditions, bloom to become the dominant ones. I have one, my most common firmicute in the red here is an oral bacteria that's 850 times the healthy value. And this is what is so cool, is to be able to know that. I mean, it's just great. And so I spend I mean, I don't know what you do for fun, but for me, there's nothing better than sitting in front of a spreadsheet with a couple of 100,000 filled cells, which is what having this many microbes times 50 people has, and then looking for patterns. I mean, that's just about as good as it gets. I can do it on I've learned how to do it on my PC. Anyway, thanks for